This is TWIS. This Week in Science, episode number 933, recorded on Wednesday, July 5th, 2023. Free the science! That's right. I'm Dr. Kiki, and tonight on the show, we will fill your head with slowing, shrinking, and hunting. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Those who fail to learn from history are condemned to repeat it. A plausible yet daunting idea, for if previous concepts of the past are any indication, the story of humanity is a tale of the damned. Humans have seldom understood the past, and histories passed down through tradition, by spoken word, carved into rocks, written down on scrolls, read from books, contemplated, debated, and taught in schools, are often wrong, misleading, self-serving, or entirely makey up. One element of modern society has attempted to put a halt to the doomed nature of humanity: science. Through anthropology, archaeology, and genetics, the true evidence of human migration refutes the endless origin stories that have circulated and shows that we are but one people with a braided stream of interconnected stories. Through population studies and medical research and epidemiology, we trace the paths of pathologies, pointing out the hazards, foretelling the risks, to understand better how to preserve life based on the missteps of the past. In geology, the formation of strange features explained. In astronomy, the yearly meanderings of stars reframed. In physics, the fabric of space and time entwined. In cosmology, the birth of our universe revealed. In every breakthrough research paper, an understanding of past knowledge leads to the current discovery. And while those who do not learn history may be doomed to repeat it, those who do learn from it are celebrated here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again. So excited to be here to discuss all the science news from the past week that we want to talk about. Of course, there's lots of news out there.
There's like 5,000 stories a day. I don't know where we can get all
fire
of it.
hose. If you really, really get into it, it's a fire hose. But we're picking the good ones. We're picking the interesting ones. We're picking the ones worth your time, everybody. So thank you for being here and sharing your time with us tonight on the show. Woohoo! We have all sorts of great news. I have news about space, life, aging, and stinky hands. What do you have, Justin?
I have a tale of people getting history wrong. I have a test of evolution and an idea that we've had on this show a few times. Potential therapy for Fragile X syndrome, and a Hepatitis C unmasked.
Unmasked? I didn't know it was wearing a mask. We'll find out.
Well, that's the whole story. Now
it's like the Zorro of hepatitis. I don't know. Okay.
Yeah.
Blair, what's in the animal corner?
Oh, I'm keeping it simple this week. I have minks, I have bees, and I have ticks.
Ooh, we don't like the ticks. We like minks. They're very soft.
I have some very interesting tick news this week.
Okay, great. And we always love bees. So, okay. all the good stories. We're excited for everything. As we get ready to jump into the science, though, I do want to tell everybody that if you are not yet subscribed to This Week in Science, so you're just jumping in right now and just hitting this part of the show, you can find us. Look for This Week in Science on YouTube, Facebook, and Twitch. We're also TWIS Science on Twitch and Instagram and Universidon, and we're out there. Our website is twis.org if you want show notes and more information about us. And additionally, we stream live to the YouTube and the Facebook and Twitch at 8 p.m. Pacific time on Wednesday evenings. That's why we're here now. Okay, time to start the science. Are you ready? Because I am going to dive immediately into the fabric of the universe.
Ooh.
Put in your earplugs and your nose plugs. You don't, you don't want to get anything out there.
Right. Well, teddy bear material. That's what I'm expecting.
Last week was apparently a huge week for big physics. The big physics experiments were announcing all sorts of amazing results. Thursday last week, there was a press conference. Researchers studying gravitational waves announced their latest results, which have been looking into pulsar timing arrays to investigate the detection of gravitational waves. And we've talked about this detection of gravitational waves before in the context of the LIGO-Virgo experiments, which we know of as these laser beam interferometers that are located here on the surface of our planet. But because those interferometers are located on the surface of the planet, they are limited to the resolution that they can get, whatever size of a wave washes past these detectors that is caught within their size frame. So we're planetary resolution limited. And researchers for a while have been like, hey, what else can we do? Well, we've got these pulsars that are out in the universe. And so maybe we can look at these pulsars. We've been studying pulsars for a couple of decades now. We've been measuring, got all this data. Maybe let's go back and dig into the data and keep measuring and see if there are any differences that would be on the nanometer scale, really. They're looking at differences or the nanosecond scale in timing of the light signature that reaches our planet. Pulsars are usually these high energy stars that have a very clock-based signature, very easy timing. We know that they're always going to burst at the same integral of time on and on and on. But the idea is if they are awash in an ocean of space-time in which there are giant gravitational waves millions of light years across, tens of thousands of light years across, that have been produced by the merging of supermassive black holes, then perhaps we can start to pick up this larger scale resolution based on nanosecond differences in how the wave pushes and pulls the Earth towards or away from those pulsars and changes the time of those waves hitting the planet, or how we detect them. And it's in nanosecond scales that they're looking at on the distance of light years, thousands of light years.
So I guess the first thing was we needed to come up with a clock that could handle nanoseconds, because that seems like, hey, was that a nanosecond or was that like a million nanoseconds? I can't tell the difference.
It's a lot, yeah. It's a bunch of nanoseconds, the amount of timing is this very accurate timing that they are using to be able to detect these gravitational waves that are crashing and like ripples on the surface of a planet or the surface of the ocean that are driven by the wind, or if somebody throws a rock on a pond, or one rock on one side of a pond and another rock on another side of a pond, how do those ripples interact and what happens to the stuff in between? This is another, the data coming through is not necessarily surprising to astrophysicists. They expected they were going to see differences. So what the results really are is a confirmation that now we can use pulsars to measure gravitational waves on a massive, massive scale with a resolution that is a very high resolution to be able to determine supermassive black hole mergers around us, to be able to determine how the fabric of space-time is expanding, to be able to do all sorts of experiments moving forward to confirm ideas about how space-time works and our place within it.
Yeah, the only thing that... Well, so there's a requirement, it seems like, on this, that the quasar pulses be accurate to within nanoseconds.
These are pulsars, not quasars. Pulsars.
Sorry, pulsars.
Yeah.
Because I kind of get like, space is really big. We're going to start at the beginning. Space is big. Okay.
Yeah.
So we have all these little beacons out there that are pinging us. Bing, bing. Now, if they're completely synchronous in their pings, ding, 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 ding. If they're keeping a very steady nanosecondy beat.
Each individual pulsar is, and so we're able to use all the different pulsars.
We can tell once they get out of alignment, then we might be able to, right? Is that the idea? Back engineer gravitational, spatial, stuffy movement over vast distances.
Yes.
That's if they're exactly accurate. Which are big things. Big things.
Right. But that's the thing, is that we have measured them so far to be very accurate. And a lot of researchers were like, well, is this going to work? And then they started putting all the data together. And after about 15 years of data from some of these places, there's the Pulsar Timing Array in, I think, Australia, and then Nanograv, who that's another, the North American Nanohertz Observatory for Gravitational Waves. There is another group out of Turkey. And so there are it's an international consortium of researchers who have all separately kind of looked at their data and gone, hey, we have enough data that this looks like it's really going to be a thing. And we can move forward with this. And because, the amount of data that they have is just, every year they get more and more and more. And it's not just one research group, it's multiple pulsar research groups internationally starting to collaborate to be able to, in the future, pool their data together and get even stronger measurements. There are questions as to whether or not, string theory, the string particles that are going in and out of existence, whether or not the string particles have an influence on gravitational waves and the movement of space-time. And this is something that we can start looking at to see how matter, dark matter, other things influence space-time. But yeah, it is ridiculous. These pulsars spin sometimes hundreds of times per second. So the metronome actually is a super quick metronome. Yeah. And that's why it can be nanosecond or picosecond kind of scale measurements.
That's super cool.
Yeah.
So
anyway.
I want to know, like, okay, well, it turns out, like, where are they going to find it? Well, it turns out that everything's moving sideways. We didn't know that before. The whole universe, it's not just expanding, it's moving off to the left a little bit.
Well, we haven't gone that far. Yeah, haven't gone that far yet. But another big question researchers have been looking into, that data was published this last week in Nature Astronomy, is a question as to how Einstein's theory of a general theory of relativity stacks up against how we see time and things and time move forward in the distant early galaxy. And we've been limited historically by supernova as our clocks, which take us about halfway back in the age of the universe. And we've been limited just by those supernovae as our clock. And so we're like, okay, well, going back about the half the time of the universe, we're not really seeing the reduction in time's speed relative to where we are right now. Because as we've seen, there have been other experiments where we've put gyroscopes on satellites, put them up into orbit around the Earth, and done timing experiments of clocks here on Earth and clocks up in those satellites just above our planet in orbit, and been able to show that there is a difference in relative time, where the time moves slower the further away from our relative position the object is. So that's time dilation, and we haven't really been able to support it as much as we'd like with just supernovae. But now these researchers publishing in Nature Astronomy have been able to show using quasars, which are a different kind of pulsing object. These are quasars that are at the center of supermassive black holes, and they are the spinning energetic the spinning energetic signatures that we can see that shoot out. And so they rotate, and their beams, like a lighthouse beam, flashes past us as that beam of energy goes past. And so they go at a kind of set speed, and we're able to get like, okay, the quasar, we can measure that speed, we know where it is. And these quasars, they can take it back to about 1 billion years after the Big Bang. And with the quasars, they have been able to support Einstein's general theory of relativity's prediction of time dilation. And in fact, about 12 billion years ago or so, 11 billion years ago or so, there was, if that time appears to be flowing a lot slower than time appears to us right here, right now.
Yeah, I don't think we needed Einstein for that, to be honest with you. Like, I'm only approaching 50. Past it, maybe. I can tell you, things are speeding up. Things
That's
are,
true.
oh boy, time is going by a lot faster than it was just some decades ago.
You need to do new things. Lots of new things. They're supposed to slow time down, I guess, to the human brain. I don't know, whatever. But the researchers, this is a very novel ability to be able to take the ticking of the quasars and apply it to a standard clock that can be used similar to supernovae for measuring the speed of time, and to be able to show that, yay, expanding universe, time moving more slowly, further away, further back from us. Woohoo! Another Einstein nail in that structure. And then the final cool thing that a project I've been following for many, many years is the Ice Cube experiment, the Ice Cube collaboration down at the South Pole, Antarctica. These researchers have been hunting for neutrinos for a couple of decades now. And in their hunt for neutrinos, they have taken the cleanest ice that they could find on our planet, which is this beautiful, clean ice, clear, with no compounds that get in the way. And they've created these detectors that detect neutrinos, which are highly, highly energetic particles, but that don't really interact with anything. So unlike cosmic rays, which bump into stuff and then produce neutrinos, the neutrinos themselves, they come in, and if they hit a detector, they don't really turn into anything else. And because of what they are, researchers can figure out where they came from. Now we have a bunch of neutrinos that we can track back to nuclear facilities here on the surface of our own planet using machine learning algorithms. The Ice Cube collaboration has now figured out how to take out the noise of cosmic rays that hit our atmosphere and then create a shower of neutrinos down onto the planet. And so they have been able to get a picture that is their most recent result that they have reported on of the Milky Way. So they are using their neutrino array. They've been able to visualize a bunch of locations within the Milky Way galaxy's silhouette that we can see. We're looking at it kind of from the side, and there are a bunch of locations that appear highly energetic. They don't know what exactly they are. They haven't pinpointed them specifically, but they do coincide more with the center of the Milky Way galaxy, and it's just a very exciting advancement. And so we'll be hopefully seeing them clean up the data a bit more, get more data on the neutrinos that they're detecting, and eventually be able to get a really interesting picture of the inner Milky Way galaxy and the most active structures within the Milky Way that go into giving the Milky Way the structure that is the galaxy we live in.
So to summarize,
yeah,
this past week we may have been able to figure out how to see the structure of gravity throughout space, the structure of time throughout space, and identified all the little neutrino emitting objects in
our galaxy. Yeah, and then once we start looking at our galaxy, then we can maybe look outward and see out of our galaxy. But we're looking at big physics this week.
That's kind of a big week.
Kind of a
lot.
I know.
Should spread it out over the year a little bit, but go for it.
I was laughing. I was like, Ice Cube. I mean, normally people would be super excited about the Ice Cube neutrino stuff, but like the gravitational waves. I mean, that's a big one, and they're both announcing right at the same time. I mean, all this stuff coming out. They're probably I think it was all announced last Thursday.
That's right.
There's only so much we can do. But yeah, moving forward, we can hope for many more amazing big physics results because...
Well, maybe not many more. That might be like... That's kind of a lot right there.
It's a lot, but I think we're on like these experiments are starting to bear fruit that, were begun decades ago and have been building and building and building. And we're starting to really see the data come through and the information come in. And so these structures and these pictures of our universe are slowly starting to come into focus.
So something very similar is taking place in medicine.
Yes.
Right now. So we've talked a little bit about prime editing, which is already showing some tests of being able to change genes within your body while you're alive, swap them out. So if you had a genetically based or gene-based variant disease, that can get swapped out curatively. Still 5 to 10 years, but legitimately 5 to 10 years, they've tested it and it works in some experimentation, right? There's base editing, which is another way of doing this. There's the universal donor stem cells. There's a universal CAR T cell therapy. There's more and more of this, like, we're on the cusp of talking about real intervention, real therapeutics, and real cures for diseases that up till now have not been treatable. And so I bring you Fragile X syndrome. May have a therapy on the way with yet another method that researchers are using to combat disease. So Fragile X syndrome is leading inherited form of intellectual disability. It's a big part of the autism spectrum disorder. Patients can present with behavioral alterations, hyperactivity, impulsivity, anxiety, as well as poor language development skills, seizures, tendency towards the lower IQ as it is a neurodevelopmental disease. So it's a genetic disorder that disrupts a single gene. Now, it's not like nobody's studied this. People have worked on this. People have tried to do stuff on this. It attacks basically one gene, FMR1, which is then named Fragile X messenger ribonucleoprotein because it's so involved in this one disease that they've been studying. Many genes, though, produce more than one product, and FMR1 does more than most. It's hundreds of mRNAs are being expressed incorrectly and spliced into white blood cells, brain tissues, in individuals with Fragile X syndrome, leading to the large swaths of symptoms and distributed neurological development pathologies. So, researchers have discovered that a large part of the problem is the splicing of the mRNA. So the little bit of mRNA gets copied off the gene, and then all this, genes get spliced into different mRNAs. They go on to then become different proteins, which then can be further modified to go on to do different tasks in the body. So one gene can make hundreds of different proteins that all have different tasks. And they're finding that it's this missplicing taking place that's the problem, and not the gene expression itself, not methylation of the gene that has been previously believed. Enter, I'm going to keep messing this up, antisense oligonucleotides, which are basically a form of engineered single-stranded DNA that can be used to bind to mRNA sequences. They bind to specific sequences based on how you've engineered them, and they can do all sorts of interesting things. They can induce degradation, so that little bit of mRNA breaks down. They can modulate splicing. They can do prevention of translation, so it's there, this gets attached, but it never gets turned into a protein. They can interfere with alternative mRNA processing events, which then can either inhibit or upregulate the production of mRNA, which then inhibits or upregulates the production of downstream proteins. Right? In this case, the researchers develop antisense oligonucleotides to form a little patch that sits there on the mRNA, and it causes the splicing machinery to skip past the improper splicing spot in the RNA. So the mature mRNA is formed without the missplicing event. They did a little bit of collaboration with some RNA therapeutic field experts, and they found a 2 antisense oligonucleotide combo successfully inhibited the missplicing, as well as had a little bit further effect. So basically, this led to the production of normal levels of the protein FMRP. The absence of those proteins defines Fragile X syndrome. So as far as the cells that they were working with in a lab are concerned, they have a therapeutic cure. Okay.
Wow. So as far as the cells are concerned at this point, the cells, they're fixing the problem. So that
Yes.
It fundamentally is saying, okay, you have this mistranslation and you're missing it, this one part. And so we're going to fill it in. We're going to make sure it doesn't get missed, and then the cells will be fine. And so now you just have to make sure that it works in mice, and then in primates, and then in humans, right? And so they have to move forward through all the clinical trials. But that's
so it's
amazing.
really interesting too. Okay, so currently artificial antisense oligonucleotide therapies are in various stages of clinical trials already across a wide range of RNA-based diseases. There's rare skin diseases to very common neurological diseases. There's a lot of potential for the artificial antisense oligonucleotides, including potentially, hopefully, a new name at some point.
Not so long. I'm sure if it becomes an injectable drug or, something of that sort, it'll have a nice, proper pharmaceutical title.
You can call it ASO now for sure, or you can go RNA therapy. Going forward might be okay.
But RNA therapy is vague. It could be.
Yeah, it's a little broad sounding. So anyway, this this is the huge part though. This is actually the biggest lesson takeaway I think from this. This is according to Joel D. Richter, who's lead researcher on this particular project. We never would have found this using a mouse model of Fragile X. The mouse model is a gene knockout. Because it simply doesn't have the Fragile X gene, there is no mRNA that is made.
Right.
We only discovered this missplicing because we were working with human cells. So, huge alert to all genetic diseases that are being studied in knockout mice. Have you checked for the missplicing in the actual cells first? Because that's turning out to be a huge thing. And it kind of actually, in a weird way, so I've heard about this antisense oligonucleotide therapy before.
Mm-hmm.
And it's being used towards some very common diseases. It's being used for a lot of extremely rare diseases that nobody's ever heard of. And now I'm starting to wonder, like, is it because the labs who were working on that, like, weren't structured or didn't have access to knockout mice, wasn't in the budget, they don't have the space, they're working on this niche disease because, hey, nobody else is, maybe we should do this. And because they had to work with actual cells and didn't have the fancy mouse knockout models, they were able to see the missplicing. And that's why there's so many of the applications for this for very, like, diseases you haven't heard of with a very slim amount of people who are even getting it. And there's a ton of therapies underway. So just add this one to that list, that stack of new interventions, therapeutics, and potential cures that are in the pipeline thanks to science and medical research.
Yeah, and that this one particular disease might be cured, fundamentally, by this as a treatment, and it could also impact a lot of others just as a methodology. Yeah, and
it's going to be interesting because this is also a developmental disease, so you kind of have to wonder, like, is there going to be a sort of reversal effect that's possible? Is this going to be something that's an early intervention that then allows regular development, so that you get the word Fragile X the early stage, and you don't have to panic?
Yeah, I mean, you'd want to get it early, I'm sure, but yeah, there's a lot more research that needs to be done to see how it, it is a breakthrough.
So there's a lot of...
We don't know. So many. Blair, I have questions about mink brains.
Mm-hmm. Fragile mink brains. Fragile.
Are they fragile?
Yeah. I mean, them, along with many other animals we have domesticated, have brains that shrink during domestication. This is a phenomenon that is well studied. And to this point, the expectation was that once you shrink, you can't go back. So once you shrink,
you're a mink. No.
You were a mink before too, but this time you're a dumb one, I guess. I don't know. But so sheep, pigs, cows, basically all domesticated animals, mammals specifically, have smaller relative brain sizes compared to their wild counterparts. But American minks appear to be special because the ones that have escaped captivity and have become kind of these feral minks in Europe, even though they're American minks, they have regained some of their brain size by being in the wild over 50 generations. Yeah, so generally speaking, these domesticated animals do not regain relative brain sizes to ancestral forms, even in feral populations. But part of the reason that that's understood is that it's really hard to study this. How do you study a wild individual, a domesticated individual, and then a domesticated turned feral individual all at the same time in large enough numbers to come up with statistically significant measurements? It's very difficult to do. Yeah. And so the American mink is a perfect individual because they were domesticated for the fur trade over a century ago. They were bred in Europe then for fur farming, and captive animals escaped. So you have wild minks in North America. You have domesticated minks in Europe in fur farms. And then you have the feral mink that have escaped in Europe. So you have three distinct populations. They are not cohabitating. There is no bleed over. And so they have these really distinct data sets. On top of that, you have the situation with mink where it's really easy to measure the brain case. Why? Well, there's museum collections of American wild minks. The fur farms obviously get mink skulls regularly.
Yeah.
But via fur procurement. And
hey,
then,
you mean they don't shave them? I thought they were like clippers.
No, you need the skin.
They take the whole skin for this, not just the fur. Yeah.
Yes.
It's not like
and
sheep.
then you also have the fact that the American minks that have escaped in Europe are feral, they are invasive, and therefore they are regularly culled. And because they are culled, because they are eradicated from the wild, they have collections of mink skulls over time from these feral populations. So you're able to measure the skull case of all of these different minks. And so they were able to calculate relative brain size related to their skull. And the brains of captive bred mink shrunk about 25% compared to the wild ones from America. But in contrast, the brains of the feral mink grew back almost all the way to the wild size within 50 generations. So there's a couple things going on. Are we misrepresenting how this works in the wild? Or are minks special? Now, minks, along with other weasels, because they're in the weasel family, as well as shrews and moles, have a process known as Denel's phenomenon, which is where they have seasonal changes in their brain size. So they actually have this kind of plasticity related to brain size built into their genetics already. So it's possible that these minks, along with shrews and moles and other weasels, could have kind of special adaptations that allow them the flexibility to grow those brains back to normal size. Or it could be that we're just bad at measuring this data because it's hard to get, and everybody has the potential to get their brain size back. We don't know. But what we do know is that this bucked the expectation of how domestication works. So
we're also looking at the issue of whether or not they're collecting. I mean, I guess they're doing a big, broad sample, but what season are you collecting the animal in? How old is the animal?
Right.
How does, age and seasonality, if we know that the brain does grow and shrink, how does that all tie into the way the measurements work out?
Right. Well, and then the other thing is, yes, they got back to the size they were at. Are they using their brain in the same way? Is it wired the same way that it was originally? So that's the piece we don't know. All we know is just based on size, they're getting the size back. But how are those brains functioning? Are they functioning like wild minks? Or are they just smarter, domesticated minks now? Did they have to write new pathways? Like, what's happening in their brain? That's way harder to figure out. But maybe someday.
Yeah, I think that's a fascinating question. What is the actual change that's happening? Even though we're seeing the size come back, what does that mean?
Yeah.
Great question. I love it. I love studies like this. This is what a neat use of all the data sets and the different ways of getting all that information together to be able to answer the question, or at least start to get at it. It's very cool. Let's see. Scientists have been looking into why aging. Why?
Yeah, why? Stop it.
Why?
We don't want.
We don't want. There are organisms on our planet. Usually they are, related to our jellyfish ancestors that we, haven't really been close to for, 500 million years or so, 600 million years. But anyway, there are lots of squishy water-based animals that don't like to age, that can almost live forever, that just keep regenerating themselves. And scientists are like, okay, we're going to look at you little squishy water hide tunicate type things. What are you doing to stay alive forever? And, how does that different from the mammals and other vertebrates who like to die? Well, maybe not like to, but do die. So what's going on there? Well, there's a process in aging, which is known as senescence, where suddenly the cells start to deteriorate, tissues start to deteriorate, and it's a period of time of known deterioration. And there are certain molecular...
It's a good thing. And it's a really good thing.
Yes, senescence is good. Apoptosis is good. Like, there are lots of these cell death processes that are really great. Like, you don't want all the tissues to grow forever. You want there to be stops somewhere.
Because once there's problems, that's you get into cancer territory and all that other kind of terrible stuff.
Yeah. So these researchers were looking into a species of of organism, little sea creature called Hydractinia symbiolongicarpus. And these are little tiny tunicate type sea creatures that are related to jellyfish, and they live on the backs of hermit crabs, and they have little tubes and little fingers that come up from the tubes, and they sway in the water, and they collect their food into their single mouth that goes into their tube where they digest it, and then they probably poop out of their mouths too, but that is not something we're talking about right now. What we're really talking about is the fact that if you were to look at these hydractinias, they have a collection of stem cells that they keep toward their basal midsection, and they keep those little stem cells in reserve in case anything goes wrong, and they can keep growing from those stem cells. The stem cells are a very important part of how they regenerate. However, those stem cells, relative to the size of these hydractinia, are far away from the mouth region. If you cut off hydractinia's mouth, it will start to regrow itself. It will regrow a whole new thing without any of its stem cells. And so the researchers are like, what's going on there? And so they got into looking at the compounds that were released from the cut area when they cut off hydractinia's mouth and what was released there. They found there were a number of components that were similar to senescent compounds in vertebrates and animals that go through aging and have tissues that die. And so this was a very fascinating discovery for them because, as they publish in Cell reports, that this leads to a realization that the processes of healing and aging are intertwined, and that possibly aging evolved as a result of healing in the first place, and that we wouldn't have aging if it weren't for adaptations and selective mutations that have moved forward because of healing and survival.
So no more healing for me.
Stop healing, Blair. That's it. Yeah, but it's
no.
Yeah, I really appreciate this study because it does kind of bring that kind of life and death, aging and birth, into this full circle of scientific inquiry and discovering that there are these connections molecularly that can be with senescence and aging that can be tied to healing and regeneration in the first place. And this may give us some very interesting pathways to go down when it comes to doing things like regenerating nerves or limbs or even living forever. Fun times.
We've back engineered a sea creature that allow you to live forever, but there's one compromise you're gonna have to make. You're gonna have to poop out of your mouth.
You have to poop out of your mouth. But we're not talking about that.
That's the only one. I feel like you also have to give up your bones.
That's right. Yeah.
Oh yeah, you don't need those. You're not gonna want those.
You have
too
to
complicated.
You have to.
We haven't figured out that.
Those fan dudes that they blow up outside of auto dealerships.
The inflatable tube guys.
Inflatable tube guys. That's kind of what I imagine.
Yeah, that's pretty accurate.
All right, Justin, tell me about minimal cells. Is this like minimal techno?
Yeah, sure.
Okay, sure.
Why not? So a real test of evolution has taken place, ladies and gentlemen. The simplest possible organism would be one that possesses only the minimum number of genes, just what it needs to survive and reproduce and nothing else. This also would make it very unstable, as any mutation could be lethal. The more efficient the model is, the less likely that any changes to it can be good. So how did evolution ever get going if life is so precarious in its sort of rudimentary state? Researchers in the Department of Biology at Indiana University decided to answer that question by taking bacteria with a small genome 901 genes in the wild type, and eliminating everything but the core survival essentials to live and reproduce. So they got a reduction of 45%, dropped the bacteria from 901 down to 493 genes. In humans, we could lose like 98% of our genes, I think, and we'd still be okay. But this one, 45% drop down to 493 genes. A smaller genome at 493 than any wild organism known to science. If there's one thing that history of evolution has taught us, is that life will not be contained or constrained. Life likes to break free occasionally, expands into new territories, it crashes through barriers.
As Jeff
Painfully,
Goldblum
maybe even
says, it does what?
dangerously. Eventually, life finds a way.
Right.
And in a lab in Indiana, life did just that. So over the course of 300 days, the sequence shorty showed off an intensive mutation rate, highest recorded of any cellular organism ever. It was somewhat expected, I guess, because organisms with smaller genomes have been shown previously to have higher mutation rates. And the bacteria they were working with already has an elevated mutation rate. So while the rates were faster than the wild type of the bacteria, it wasn't crazy faster, just faster than the wild type. It was allowed to evolve freely for 300 days and then tested along with its streamlined version and the wild type. Essentially, the wild type bacteria in the streamlined version are placed in the tube. The wild type takes over. Good data point to the discussion that we've had a few times on the show about why life doesn't seem to de novo be starting from scratch on Earth over and over and over again. Once niches are filled, there's no way for new basic genomed life forms to get started. They lack the attributes to compete. So the wild type versus the completely shortened version, the wild type just took over the tube. The shortened version didn't make it. Now, in the wild type versus the 300-day evolved strain, the evolved strain held its own pretty well. So life finds a way, and quickly. Through 300 days of evolution by itself, the bacteria technically went through about 2,000 generations, which would be 40,000 years of human evolution if you calculate it that way. Though based on population and mutation rates, that small 493 gene genome, a new mutation would hit every nucleotide in the genome more than 250 times during that 2,000 generations. So,
Ooh.
a lot of opportunity to find out what works and what doesn't.
Yeah.
In that experiment. So, along the way, the researchers uncovered genes, proteins, and traits that are critical to evolutionary performance of bacteria with the smallest genome of any organism grown in pure culture in the laboratory. In what researchers are calling the ancestral state, the working approximation of minimal cells had significantly reduced fitness with less than the 500 protein coding genes that it was allowed, and very few redundancies. So, theoretically, it could have, maybe should have, just gone extinct. Instead, what they witnessed was natural selection during extended laboratory growth outweighing any deleterious effects of genome disruption and drift, leading to a more robust form within a year.
I think that's very impressive. That even with the minimal amount of genetic material that was there, that wasn't like a constraining factor. I mean, it's a bit constraining in that there's not as much genetic diversity within the genome to begin with, but the fact that there is mutation happening, single nucleotide mutations, there are insertions and deletions over those 300 generations that impacted it enough so that the fitness increased, it was able to compete. That minimally evolved ancestor, or that minimally involved cell, that version of life, bacteria, even though it had just enough to survive, it was able to change and compete. And that should give us pause when we think about where life came from in the very beginning.
And it's a great, I think it's a great example of, what we talked about why life doesn't keep showing up over and over and over again, because, you can't compete when you don't have anything but the ability to eat one thing and reproduce. Right? There's somebody else has got that niche. Somebody else is going to fight you for it, and they got skills. The other one is that this didn't have, like you were just saying, this didn't have a population with which to keep like, oh, you're different than me. And then at first, it starts, they're all starting the same. And it's mutation evolution completely ran the show in the beginning. And then maybe towards the end, now you're like running into other successful versions within the population that are becoming this more robust form that's able to survive. But it shows that mutation evolution and selection from that is very powerful. Actually, much more powerful in those early stages. Yeah.
Yeah. That those early stages, as long as there's enough there, then there is enough to grow and compete and to, I think it would be interesting to see how much longer does it take? How many more days of evolution would it take on the minimal cell to get to a point where it can outcompete or really, really, truly compete fitness-wise with the non-minimal cell, the original wild type? And basically, how long would it take? That question with the mink brains, right? How long would it take before it got back to that kind of wild type version? And when it did get back to the wild type, is it just like the wild type? Or, what are the differences? And we can see that filter right there. I think that could be cool.
Yeah. And then that's the other thing is like these didn't have any competition but each other, right? So they're like, we're just going to grow and see who's better at growing in this space using the same resources. Still probably a pretty friendly competition compared to going out into the environment where there's all these other strange bacteria and viruses and creatures that are out there trying to get you. I don't think it would have done as well there. I'm just guessing without any exposure to any of that before. Probably would have failed even worse. But did really good holding its own against its wild type, more advanced version of itself, I guess.
Yeah. And I think this pulls right into my last story for this first part of the show here. Research out of MIT, the Massachusetts Institute of Technology, published in the Proceedings of the National Academy of Sciences this week. Chemists at MIT were focused on bacteria for their paper that was just published looking into photosynthesis and the efficiency of photosynthesis. And in their study, they were looking at the photosynthetic reaction center, these light harvesting proteins that are involved in the light harvesting complex, to figure out how, efficient photosynthesis is in plants and in bacteria, and why it's so efficient, and why can't we do photosynthesis as well? Why, what do we need to do to make it work well? And very often when we're putting together our photoelectric diodes and the solar panels that we're constructing, everything's in a crystal lattice. It's all neatly ordered. The crystalline structure allows the electrons to flow very easily. It all makes sense. But the big news from this study is that when they tried to control the size of the membranes and the distance between the proteins involved in the light detecting, the light gathering structure in these photosynthetic purple bacteria, they found that when they made it nice and ordered, that wasn't as efficient as when they just let biology be messy. And so photosynthesis was better and more efficient with disordered, just wherever it goes kind of placement of these proteins within the membrane. So that's
awesome. That's like entropy is the plan.
Yes, it kind of, but it's really the researchers themselves commenting on this study. They say ordered organization is actually less efficient than the disordered organization of biology, which we think is really interesting because biology tends to be disordered. This finding tells us that That may not just be an inevitable downside of biology, but organisms may have evolved to take advantage of it. Evolved to take advantage of disorder, right? Yeah.
Why bother folding your clothes? Just throw them in a pile. You're wasting your energy.
Disorder. Your disarray is going to make your life more efficient.
I've seen a lot on social media lately about doom baskets. That's exactly what this is about. Like, why are you putting all this energy in to creating order where it's not required? If it's not required, it's a waste of energy.
Right. And that is specifically, the chemical aspect of what they're looking at and the thermodynamics of it, or the, how does the energy flow? That's right. Disorder. It's better in biology. Hee hee. Which I think is so much fun. Well, this disordered TWIS program, we've been here for you, and we're so glad that you are here joining us for another episode. If you're enjoying the show, please tell a friend about TWIS today. Oh, and while you're talking with your friend about TWIS, maybe take some time to head over to twis.org and click on that Patreon link. And then you can choose to support the program moving forward, because we are listener supported, and we would really very much love your support. $10 or more per episode, and we will thank you by name at the end of the show. Additionally, $15 and more, and you'll get a new sticker with TWIS art from Blair, fun things from TWIS moving forward. We hope that you do decide to support us. We can't do this without you. Thank you for your support. All right, it is time to come back now to that part of the show, that part of This Week in Science that is filled with animals. Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I got the buzz on bee homes.
Oh, bee homes.
Yes.
Wait. Okay.
So
hive, yeah?
There are hives. That is a thing. Many bees and wasps live in colonies of several thousand individuals, but there are also solitary bees. So when you see things like bee hotels that people make, these are usually for solitary bee species, or for bees who don't have a colony or a hive of their own. Excuse me. One of these solitary bees is the horned mason bee, Osmia cornuta. Probably saying that wrong. They choose a nest site. They usually will use existing cavities that can be abandoned nests of other bees. It can be wall cracks. It can be artificial nesting aids like the bee hotels that I mentioned before, which are usually, it could be, pieces of bamboo or little wooden dowels that are hollowed out, any number of things. But they like their little nooks and crannies. But the question is, how do they know what their cavity is? How do they know which hole is theirs? When they go out and they pollinate and they munch on nectar and then they come home, how do they know which hole is their home? And so previous studies have suggested that it's all about vision. They recognize it based on how it looks. That one's mine. It's how we usually tell.
Yeah,
mine is the blue house
pretty good vision, right?
With such and such number on it, and the mailbox shaped like an elephant. That's my house, right? So I'm just manifesting for my future house. Anyway, that was the previous studies. They all said that it was based on vision, but researchers this week, they were skeptical. They thought there was something else going on. And they did chemical analysis and they did some behavioral experiments to show that bees also use olfactory cues at their nest entrances. They have these olfactory profiles. It's the same as they have on their cuticle, which is the outside of their shell. And so when they got rid of these olfactory cues on their hotel rooms or homes or whatever you want to call them, the bees became disoriented, really had a hard time finding where their house was. So olfactory cues are an important part of this, not just visual cues. So they're using multiple senses for orientation, and most likely this is not the case just for this one species of bee. It's probably true for lots of solitary animals that find homes and claim them as their own. And so, of course, this has implications for protection and support of bees, which we're always talking about. They're pollinators. We got to save them. So we want to make sure we're not accidentally masking the way that their homes smell. If we have our bee hotels in our backyard, you don't want to be spraying those down all the time,
Right.
changing the way they smell, any of that stuff. But that also means that there's more study to be done. They want to find other solitary nesting Hymenoptera, which are bee-looking things. Is the scientific way to say that. So they want to find other solitary bee-looking things and see if they also do this and use the same cues, because chances are they do. And then how can we help maintain those populations? But also, if they're pests, there are also hymenopterans that are pests. So if you have a pest, is that something that you can kind of weaponize, knowing how they find their home?
Weaponized home finding. Yeah. Well, I mean, it'd be like if you want. Yes, I'm hiding. It would be as if you want a bee or a what is it, a hymenopteran, solitary hymenopteran, to take whatever, I guess, pest management substance back to its home to kill its young, or like how would we want to get rid of the murder hornets, which I believe they don't have large hives, but
yeah, or just also if you wanted to discourage the murder hornets from being in your backyard, if you could somehow mask the olfactory cues around where they were living, then that might discourage them from hanging around, right? So there's some kind of low-hanging fruit involved, and there's some more complicated stuff for sure. Right.
Well, we know a lot of wasps and other hornets, bees do use visual cues. So if they're, I don't know, this gets at what is most important. If the olfaction is the only thing, can they, even though they might recognize it visually, can you just confuse them just enough so they just don't go home and they don't get back there?
Exactly.
Yeah. Just enough. So, just provide just enough doubt.
I kind of like the idea, though. It's like, just get them out of my backyard. Well, they're going to go somewhere. And your neighbors are going to have the murder hornets. Larry's like, yeah.
Only if they have a good hotel for them.
That's right. Do you have a solitary pest hymenopteran hotel? Is that what you're putting together on your property?
I actually see that. I see it the opposite. I see this as being a form of bee theft that will take place. So this is a real thing. Like in farm country,
yes,
we have California has 85% of the United States bees come to California at some point for the massive pollination season.
They're trucked.
So they're trucked in from all over.
Yeah.
They're contracted out. And so you have these beehives that are set up all over on farmland, these little boxes. And occasionally there's bee wranglers, or bee rustlers, who will go and steal people's bees.
I believe they're called beekeepers.
No, but the ones that they're rustlers, yeah.
Their beekeeping suit is just in all black. Is the difference?
Yes. They have a little mask over the screen at the bottom so that nobody can. And so then, how much easier is that job if you make the really super attractive smelling home signal in a bunch of empties or whatever, and then all the bees are like, I guess that's where we're supposed to go. And then you got yourself a bunch of free bees.
Potentially. So just a reminder, though, that this is solitary bees, right? So this is not our hive bees.
Yeah.
So at this time, based on the science we have, that would not work on the honeybees.
I see a black market coming for bee wrangling or bee rustling. Bee rustling.
Bee rustling. You bee rustling. I can't go any further.
I don't know.
Blair.
But my other story, the story I was really excited to bring tonight, is kind of a weird deja vu for last week. So last week, for those of you that last time on This Week in Science, for those of you that were not here last week, for example, Kiki, I'm going to catch you up. I did a story about C. elegans, our nice little nematode wormy thing. That can hitch a ride on migrating or just on insects via magnetic fields. So they can jump crazy fast. Just they hitch a ride on the electric field between them and like a bumblebee, bop onto the bumblebee, and then they can hitch a ride on the bumblebee. So
C. elegans.
Yes.
Yeah.
Little nematodes.
Yes.
Yeah.
Yes.
And it's like nightmare-inducing leaping ability.
Yes. So, if you thought that was a nightmare, Justin, I have something even better for you this week.
Oh, good.
It's ticks jumping via static electricity.
No.
Yes.
Oh, no.
So this is a study from University of Bristol, and they wanted to see how ticks are getting on to individual animals because they don't have a physical mechanism for jumping. So they can't jump. So how does the tick get from the tall grass onto your leg or onto your dog or onto the rabbit or whatever it is, unless they're brushed up against very specifically, right? And if they were waiting for that, they probably wouldn't be very successful parasites. So the mechanism, these researchers believe, for how they're able to make contact with hosts that are beyond direct touch is static electricity.
Ooh.
Yes. So this is, to the researchers' knowledge, the first known example of static electricity being implicated in the attachment of an animal to another animal. And so for any of us that had shed carpet growing up or played with balloons in our hair or had would shuffle around in our socks, we're very familiar with electrostatic charges and how they work. But this also happens to animals in nature when they rub against grass, sand, or other animals. And that charge can actually be pretty high, equivalent to hundreds, if not thousands of volts. So that's more than you would get from, don't do it, but more than you would get from sticking your finger in a socket. So it can be, it's short-lived, but it can be a pretty strong electric charge. And so researchers wondered if the ticks could be lifted through the air via electrostatic attraction onto the animals. So for starters, they used statically charged rabbit fur and other materials. They brought them close to the ticks, and they wanted to see if they were attracted. And so this is what you're about to see if you're watching the video. They were rapidly pulled through the air across air gaps of several millimeters or centimeters. They say it's equivalent to humans jumping up several flights of stairs.
Yeah.
By these charged surfaces.
Oh, gosh.
So then they wanted to look further.
I'm just thinking it's not jumping, it's being attracted to the top of the stairs.
They were sucked. Yeah.
Suddenly, you're pulled.
You're pulled, absolutely. You're yanked.
Now, but it's not just, like, there is, in that video at least, the little tick puts up its little forearm limb thingies, legs, whatever those are. I don't even know what's in the front of a tick.
Legs.
It
They're
like,
arachnids,
puts them,
so they
it like raises them up right before, so it must be sensing the pull of some, in some way.
Is it, or is it bracing itself? Right? So anyway, so then in this next one, this is what Kiki's showing now, they use the previous measurements of the typical charge carried by the rabbit fur to mathematically predict the strength of the electric field that would be generated between an animal and grass. So they were actually wanted to see kind of what would naturally happen by an animal brushing through the grass and creating static electricity. Then they then placed ticks underneath an electrode with an air gap in between, increased the charge of the electrode until the ticks were attracted onto the electrode. And by doing that, they could determine a minimum electric field strength that they needed. And it was well within the order of magnitude predicted by their calculations. So it is highly likely that ticks in nature are being attracted onto hosts by static electricity. So what can we do with this information besides never go outside again? For me, I work in parks now, so that's not an option. And in fact, I'm hiking through tall grass pretty often, so I can't not go outside. So knowing about this,
what can you use? You could take a dryer sheet and rub a dryer sheet all over your clothing or your hair to reduce static.
There's also an entire field now of research available for anti-static spray that you could apply to yourself, to your dogs, to farm animals, that you could try to reduce ticks getting on you. But this is also
a way you could like drag a grounding wire behind yourself.
Yeah, like those cars that you see, they've got the little thing that's touching the road.
Or like when you're building computers, you always want to have the anti-static mat with the, you're grounded so that the static doesn't build up when you're building your computer.
Yeah. But I think also this is just a really good educational moment to explain to people why they have to wear long socks and they shouldn't wear shorts when they go out in the grass, even if they're walking through grass that's not as tall as their socks, right? So I got my high top hiking boots on. I'm not going in tall grass. Well, guess what? If you're walking through grass at all, you're creating a static charge, and therefore ticks could get sucked onto your legs no matter what you're wearing. So if you have long socks and long pants, you are infinitely less likely to end up with a tick in contact with your skin.
Right.
So if you understand the mechanics, it's not just can a tick reach me, it's I'm physically creating a charge that sucks ticks onto my body.
I always imagined ticks as existing on tree branches and that you walk underneath the trees and they drop on you from the sky.
Absolutely. They're like parachuting down. So the other thing
onto you.
is that this likely also applies to other parasitic species. So does this happen with mites? Does it happen with fleas or lice? That would be excellent to know. If you can spray your hair down with anti-static spray when you have a lice breakout in your child's classroom, will that reduce the likelihood that you'll end up with lice in your home? These are all really great questions that are kind of, the door has been opened to look at that. And the next step for this particular research team, to your point, Justin, is to see whether ticks are capable of sensing the approaching electrostatic charge of their prospective host and if they alter their behavior at all. So is that hand movement because they're sensing it and they're trying to catch it? Is the hand movement because they're sensing it and they're trying to brace themselves? Or is this just, do they get thrust upward and there's no choice in the matter whatsoever?
Yeah, the way it looked to me, my interpretation was that it was kind of bracing for the move, but getting ready for the landing. Like they're like, okay, I am ready. Let's go.
Right. Yeah. So do they know it's coming? That's a great question. Yeah. So
they don't know, but they know.
Yeah. Hopefully we'll have more on that later. And so that is my second week in a row of electrically charged jumping organisms onto other organisms. I love it. I can't believe it.
This is a whole area of study, the static electricity realm, just electricity in the air on us between, there's so much that we haven't looked into. Oh my goodness. I love it. Justin, what do you have?
I have the entire history of evolution to the origin of the human species. It's
Sounds like a long show. No.
Yeah. It's a long story.
Yeah.
But this has been rewritten in just the past few years, and likely we'll go through a few more rounds of revision before we approach something resembling the actual past. So in the latest version of what we thought we knew but were wrong about, archaeological evidence from across human history, a little bit going back into prehistory, is challenging the whole men hunted and women gathered trope. Turns out, men hunted and also women hunted. And men also foraged.
Shocked.
Yeah.
What? There was better division of roles and...
There wasn't any. Yeah. So, like...
Everybody did.
Well, part of it's always been a little bit silly. Like, you know, men hunted and women gathered has always been like... The part that actually used to throw me was like, so men walked past a bush covered in berries and went, oh, I wish I knew how to pick those. Too bad I can't. I gotta find a woman. She'll know how to get those berries off of that bush. So part of it's just never made any sense whatsoever. But I honestly took for granted the thing I'd been,
Justin,
told that the...
I've been thinking about it though, as the men are out hunting and they come across the berries and then eat them themselves and don't share them with anybody.
Oh,
the women go
They're
out.
like, I crave sustenance because I'm doing this hard hunting work. Never mind the women who are raising all the children. That's easy.
Bringing it home.
Yeah,
they don't bring it home. They're bringing the hunting provisions home. Only the women
they can't eat
share
it all.
the foraging provisions. Yeah. Okay. Sorry to interrupt.
No, that's perfect. I'd never considered it that way. So researchers analyzed data from 100 years of 63 foraging societies from around the world, including it looks like everywhere. North, South America, Africa, Australia, Asia, the Oceanic region. I don't know what got left out. Women hunters were identified in 79% of the analyzed societies, regardless of their status as mothers. Hunting by women, found in the research, appears to target game of all sizes, but most often, large animals. Analysis also did reveal that women were actively involved in teaching the hunting practices, often employed a greater variety of weapon choice in hunting strategies than men. There was some division of sexes involved in that women were more likely to hunt in groups, whereas men were more likely to hunt either with one or two, like solo, or maybe just with one hunting partner. Women were also much more likely to hunt with dogs, suggesting
interesting
that dogs may first have been a woman's best friend before a man.
Interesting.
Yeah, so also findings suggest that women were skilled hunters, had an interest and a very involved instrumental role in the practice. Authors note that stereotypes about gender roles have influenced previous archaeological studies, with some researchers reluctant to interpret objects buried with women as hunting tools, but rather ceremonial
of course
offerings.
Right.
Or the other way around, identifying female remains as males based on weapons or hunting tools being in the grave. So they call for a reevaluation of a lot of past archaeological evidence, cautioning against misapplying the idea of men as hunters and women as gatherers in future research. And one of the things that I also kind of find interesting is one of the theories of language that has sort of been offered out there from time to time was the importance of communication in an organized hunt. For a group with an organized hunting strategies to be able to coordinate and communicate all of what's going on during that hunt. And if it turns out that men were hunting by themselves, didn't really need to develop a whole lot of communication skills. But if women were hunting in groups and with dogs, there was all sorts of communication and signaling and everything
with
else.
men, and maybe they're all hunting together, right?
Yeah, but it might mean that women are also not only in charge of they're responsible for the domestication of dogs more so than men, but also maybe more heavily involved in the invention of communication.
You're saying women are better at communicating than men?
No, I'm not. I'm not saying they're better. I'm saying that it may have been their idea. Yeah, all along.
So, where did this idea originally come from, that men hunted and women gathered? When did this happen?
That's obvious. That came from men.
Yeah.
Yes. Anthropologists.
Well,
like white men in tweed jackets with elbow patches.
No. But they came from before tweed jackets, when men were wearing nothing but togas.
Oh, got it.
Who started these ideas?
Those guys.
These are pretty old ideas. But one of the interesting things is, along the theme of not knowing history, almost everything that we think we know historically turns out to be absolutely makety-up nonsense, passed through some filter of one or two historians who are like, this is probably how it was. Sure, why not? I'll write it down and people will repeat this for the next
because the patriarchy
years.
Or the Bible or
years.
any number of things. Yeah.
Somebody wrote it in a book, so now everybody has to believe it. Yeah. It's kind of how the power of media was once upon a time. My final story for tonight. This is researchers at the University of Copenhagen have figured out how hepatitis C avoids the human body's immune defenses, so that it can persist, go around, kind of seems to go away, and then comes back with another chronic reinfection. And there's a few people who've got hepatitis C, apparently 50 million people worldwide, something in that ballpark. So it causes inflammation, scarring of the liver. When you have damage to an organ, you need to repair it. And every once in a while, those repairs go wrong, and then that leads to cancer. So it's a terrible, awful disease. Evades the human immune system and spreads throughout the body. But how it does it has been eluding researchers, despite the fact that it is one of the most studied viruses that causes a pathology on this planet. So, University of Copenhagen and Hospital, which I can't pronounce despite my youngest child being born there. Child is where your son you were born in the hospital. That's the best he'll ever know from me, at least. Anyway, they teamed up and they discovered the virus just puts on a mask. By donning a mask, the virus can remain hidden while making copies of itself to infect new cells. The mask cloaks the virus in the form of a molecule that is already in our cells and everywhere. So it disguised by this molecule, our immune system is confused. The virus looks like it's something harmless, so there's no reaction. The camouflage being used by the hepatitis virus, the mask, to hide in our cells is called FAD, a molecule composed of vitamin B2 and ATP. So FAD is vital for cells to convert energy. The FAD molecule's importance and ubiquitous familiarity throughout our cells makes it ideal camouflage because it's conserved to the point where it's ignored entirely. It's on the team. It's on team body so much so that there's no checking of credentials of FAD at any point in our immune system. The researchers turned to Arabidopsis. Okay, boom. We're now at this teeny tiny little plant that's been researched, probably the best researched plant genomically out there. There's an enzyme for Arabidopsis that is known to be able to split the FAD molecule in two. Using the enzyme, The researchers were able to split FAD and prove that hepatitis C virus was hiding inside. What's really interesting is that this is despite all the study, this is the first time it's been seen. And it's likely not the only virus that's using this technique. So there's a lot of viruses that hide out in the system that are chronic, that last, or super long time, which is kind of weird because viruses kind of infect other cells to exist. So how can it just be kind of disappearing and then reappearing? They find it basically it's kind of wearing it like a mask. Almost sounds like a hat. Like one end of the virus is being covered by the FAD material, and the other one's out there looking for other cells to infect.
But as long as it's wearing the hat or the mask, there's no reason for the body to think that it's weird at all. And so the body goes, hey, you're cool. You're part of the whole system. I like your hat. I have a hat just like it. We like it. Can I borrow your hat? And so then they connect with the hat and bring it on in, and then hepatitis C gets access and can be on in there, right?
This is a quote from Jeppe Winter, one of the researchers on the project. All RNA viruses have the same need to hide from the immune system, and there's a good chance that this is just the beginning. Now that we're attuned to this trick, it opens up the possibility of developing new and perhaps improved methods of tracking and treating viral infections in the future. So, medical science, one more thing that we're probably not going to have to worry about. Hey, look, it's the whole thing with the kids tomorrow, right? They're going to look back. People had viral infections? Genetic-based diseases? How do you have a genetic-based disease? That doesn't make any sense. Just fix it. And you put it in the thing, you take the gene editing, and then you just fix it. Gosh.
Just fix it. We'll do everything. Easy as pie. I love the idea, though, that this is a way to potentially look for other intruders. How many hats, how many masks are out there that are essential to cell function that become, a costume that allow bad things to get entry and not be attacked by the immune system.
Even if the same one could be used by other viruses. They've just now, as we are speaking, and maybe last week, discovered that this is a place where a virus can hide. And
we don't want that.
Could be all of them. Could be all of the chronic viral reinfections are all hiding in the same place. It would be it's a great hiding place if you think about it.
Hide all over. They could be everywhere. I mean, this could explain a lot of things for diseases and various factors. Going back, I've got a couple of stories to end this show on. Blair was talking earlier about the sense of how bees find their homes. Possibly, we're talking about recognizing different things. There have been some great stories throughout the show. You were talking, a lot of things that we were discussing, but I have some fun stories that kind of fit in with these already. Publishing out of Florida International University in PLOS One, researchers have done a small study. It's not a huge study, although the headlines would have you think it's a huge study. But these researchers are investigating how the scent of your hand, or the scents that are left because of the odorant molecules, the bacteria, the sweat, the sebaceous secretions that come off your hands that you leave around behind you, how easily they can identify individuals. And this is for purposes of forensic science. For
by
being
the
able
hand?
to use, yes,
or
the hand.
Or by the scent the hand leaves behind.
By the scent the hand leaves behind.
Okay, got it. Okay.
And so in this study, they got people to hold on to little cotton things and leave their sebaceous secretions and hand leavings all over the cotton padding. And then they put them through mass spectrometer to determine what was there. And then they started putting together a statistical model that could help determine who was leaving what behind. And in the process of what they showed with their multivariate regression modeling, they were able to, based on 18 individuals within 60 that had been sampled, 18 of the individuals had stated whether or not they were male or female. So we're just looking at these two genders specifically, and they had gone through and gotten data with the mass spectrometer of all the compounds that had been left behind by the various individuals on those little cotton pads. So we've got all sorts of odorant molecules like octanol, tetradecane, linalool, phenol, tridecane, pentadecane, like all sorts of naphthalene, all sorts of things left behind from people's hands. And instead of looking exactly at certain compounds, they started doing this multivariate regression, just saying, okay, we'll just use a couple of these factors. And then once we add in a third factor, let's see where we can go. And they determined that with just 3 factors, they were able to tell specifically between males and females in the grouping, that male and female hand scent prints are completely recognizable. That when they started diving into the samples, they provided very, very identifying gender-based fingerprints.
Because hormones?
Because hormones, probably.
Is it? Or and it's, I'm going to speculate before you tell me if we know.
We don't know. They didn't look specific. They didn't have enough individuals.
Justin's gonna paint with some broad brushes related to gender now.
I got it figured out. I've got it 100% figured out. This is easy. This is
hold on. Side bet.
Whether or not they wash their hands.
Yes, no. That's exactly what I was gonna guess.
No, that's not what. Well, okay, yeah. So that, and this is very much just based on my experience with all humans, every single one of them. I don't know a single man who uses hand lotions, or puts lotions on their faces, or puts lotions on their skin, or uses a moisturizer, or does any of that. But I don't know a woman who doesn't.
I know a lot of men that use hand lotions
I know some
because they work
with
their hands, and their hands are cracked in a mess, and so they use hand lotions.
Okay, so
there can be some. I'm not saying
one of the things that they did in this study was they
with a broad brush.
They made sure people did not wash their hands for 1 hour before the study. But I do not know if they told them not to put on any lotions or anything of that sort.
Well, and do they use scented soaps or any of these other things? But yeah.
So,
but they were able, regardless, these individuals, 9 of males and 9 of females, 18 individuals out of 60, they were able to, with like a 96, almost 97% accuracy, identify the gender just based on compounds that were present. They didn't break it down to specific compounds, and that would be the next step of the research, is to actually start putting together, quote unquote, a fingerprint of the hand scents. But and this is with people who identify specifically as male and female. So we're not even opening the box of the diversity of gender identification and how this could change what would happen if you'd actually look at things more complexity.
Right.
And the chat room is completely correcting me. Of men in the chat room, or husbands who use lotion.
Lots of lotions.
It's apparently just me. However,
it's just you.
I'm now jumping onto Blair's side bet. I want to go that route with the hygiene, because there's something that I hadn't noticed before, but I have an antiperspirant, underarm deodorant kind of thing that you put on. It's in their cabinet. And I, for the first time, noticed that it has 48-hour protection. Is listed on there. And I realized, like, the only reason you would need 48-hour protection, antiperspirant, is if you don't plan on bathing for 48 hours. And then I'm thinking, I wonder how often somebody's like,
shower. That's not too bad.
How many folks look at that and go, this will give me another 48 hours on top of the 48 hours. Because I think Blair's hygiene hypothesis.
No, I didn't actually take that seriously. I thought that's where you were going. But what I was expecting this story to be about was about exactly what you said, Kiki, like a fingerprint, right? Like being able to use it in court to say, like, your hand smell is all over this crime, right? But if they've only so far figured out male, female, that's not, because that's the first thing I thought of is, like, if you change your diet, if you change your soap, if it's hot versus if it's cold out, like if you're a woman, like where you are in your hormone cycle, like all these things could impact the chemical composition of what's going into the sweat that you're releasing on your hands. It seems like it'd be super variable.
It seems like it would be, but apparently with this small sample size, and people who identified specifically male or female, they were able to get that discrimination very strongly in their data. So, could this potentially be used down the line for forensic analysis of crime scenes? I mean, at this point, it's probably better to use the scent detection of a dog, at a crime scene, looking at, you left behind a scent. Okay, dog, Fido, figure out which of the people on the lineup smell like this, that's probably a more highly accurate methodology than using this at this point in time, and probably easier than trying to wipe down for sebaceous secretions and take it to a mass spectrometer to get the data to be able to do, there's a lot there. So, yeah. John Hogan, I don't know how many people like shaking sweaty hands. I'll just leave it at that. But yeah, my inside joke to myself about this whole story, though, is like, is this giving another meaning to the phrase "smell my finger"? Anyway, just smell my finger. It's fine.
What was the first meaning?
Yeah, where was that finger? What was the joke?
I don't know. It's something. I'm sure it's something preteen.
You're saying it's another
children.
meaning. I don't think I understood the first run at "smell my finger."
All right, that's why I should just keep these inside jokes inside, and just keep them inside the Kiki brain. Speaking of brains, researchers are trying to make monkeys smarter. Well,
time.
The end goal is, it's great. We got to make all the monkeys smarter. Really, though, the study put together by researchers at Yale and University of California, San Francisco, published in Nature Aging this last week, is attempting to investigate molecular components of our metabolism that might be involved in healthier aging, potentially trying to find treatments or new drugs or supplements that could lead to improving memory, especially as we know memory is supposed to deteriorate as we get older, especially if we're looking at neurodegenerative diseases. Are there ways that we can hold that deterioration off and keep memory strong and cognitive function going at youthful levels for lifelong? They used a compound called Clotho, who is also known as the Greek goddess of fate. In this molecule, this protein is produced by the kidneys and it circulates in the blood. It doesn't cross the blood-brain barrier, which I think is really fascinating, but in multiple studies has been shown to impact memory and cognitive function in mice, in worms like C. elegans. Don't think it has anything to do with static electricity, but somehow Clotho, which is produced in the kidney, it helps keep us healthy, and it impacts factors that then go on to impact the brain and boost cognition. So these researchers wanted to know how Clotho influences primates, not just mice. So they started with mice and injected the mice with Clotho, and they showed that they were able to get a change in using rhesus macaque Clotho that had been isolated from kidneys of rhesus macaques. They showed that they were able to increase the brain activity of the mice with an injection of Clotho, and also improve the performance of the mice when they were choosing between two arms of a Y-shaped maze. Very simple tests, but they showed that there was an improvement in the mice. And so they were like, okay, it works in the mice. Great. So now let's take the Clotho that we've isolated from the kidneys of the rhesus macaque and actually give it to rhesus macaques. And in giving it to the rhesus macaques, they then showed that they were able to get upregulation and changes in the serum levels of Clotho in the macaques. And so they showed, this baseline level of 10 micrograms per kilogram, that's great. That's working. That's what we're going to use. And so they moved on to doing behavioral tests on these rhesus macaques after they had injected them with the Clotho, and they showed that the rhesus macaques actually improved on a test of memory. So they had a very simple test of memory that the macaques had to determine where some food was in a tray. They cued them and then tested them on this test, moving on over a month or so, and they showed that even after 2 weeks, even though the Clotho deteriorated in the body, in the serum within about 48 hours, the impact on memory and cognition lasted at least 2 weeks, which was how long they were testing the monkeys. So the monkeys, the macaques, they got a little brain boost from a little bit of Clotho. Now, will this work in humans? We don't know. Will it even be something that will be useful in humans? We don't know.
I'll try
How does it work in the brain?
I'll try it.
We don't know.
I'll try it.
You're like, I'm in.
I'll be the guinea pig. Anything that could make me smarter. Like I'm desperate to be smarter than I am. Like I really
your memory, right?
Oh, if I had a memory, I would be what's the word?
Just a memory.
Could make me more like a word that means like better, but in a larger. I don't know. But that's why I need it, because I can't remember anything.
Yeah, and so the question is, what kind of dosage could be used in humans? This is rhesus macaque Clotho. Could we isolate it from humans, and would a similar dosage work? Is there an impairment of function at a certain dose level? So there are lots of questions about Clotho, and I mean, honestly, it's how does it work? Why does it work? What's actually going on there? Like, why impact Clotho? I mean, it's one target, but it doesn't even cross the blood-brain barrier. So, I mean, is it, if you impact Clotho, and does it have just better general effects just for health? Does it impact other things than memory? Is it just generally good? Why don't we target? I mean, I guess it's easier than targeting something within the brain and trying to get past the blood-brain barrier. So that's, an aspect of, it would be an easier application of a drug if that's what they needed to do. But yeah, a boost in the memory of macaques.
So, but it's I'm sorry, generated in the liver?
In the kidneys.
In the kidneys, okay.
Yes. Yeah, it circulates in the blood. It was discovered in 1997 at the National Institute of Neuroscience in Tokyo. And originally it was determined that mice who were knockouts, who had no Clotho, had a lot of aging. They had like what they called a syndrome that resembles human aging, early onset disease, heart disease, cancer, cognitive decline, organ failure. all sorts of things. And if Clotho was reintroduced to those knockout mice, then they lived longer. And the Clotho was actually able, at the doses they gave the mice, they was actually able to improve their life and extend the lifespan above normal mice. So,
oh, hey Blair,
yeah,
you wanna join me in a completely unstudied experiment where we see if I can get smarter and you can live longer?
What do you think about it?
she's like, I'm making a person right now. Come on, I got other things.
2 years ago,
Yeah.
yes.
If
you had asked me 2 years ago, the answer would have been yes immediately. Okay. Yes.
Now she has to think for two.
Yeah,
it's what you gotta do. But yeah, we'll see. Clotho, are you going to be there for us as we age to improve our memories? Who knows? Might be something that's included in a nutrient supplement pack sometime soon.
Unregulated, of course.
Unregulated, exactly. Yes. I think we've done it. Have we made it to the end of the show?
Oh,
we made it.
Yeah, we made it through, everybody. We made it through the wilderness. We made it through the science. And we've come out the other side into the afterlife of the show. Dun, dun, dun. And it is time for me to say thank you. Thank you to everybody who has been a part of putting the show together. Thank you to all of you who are in the chat room right now, over in Discord, here in YouTube, Twitch, Facebook, wherever you are. Thanks for joining us and for chatting. And I want to say thank Fada specifically for your help on social media and show notes. Identity4, thank you for recording the show. Rachel, thank you for editing the show. I'm having all sorts of things right now. Gord, Arnlore, others, thank you for monitoring those chat rooms. And of course, I would be remiss if I spent a day not thanking our Patreon sponsors. So, for July, I would like to thank these TWIS patrons: Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, P.I.G., Stephen Albaran, Dale Myshak, Stu Pollack, Andrew Swanson, Fredis104, Sky Luke, Paul Ronovich, Kevin Bearden, Noodles, Jack Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Clemmy Day, Bert G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Rick Ramus, Philip Shane, Kurt Larson, Craig Land, and Sue Oster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you all for your support on Patreon. And if any of you out there want to join us on Patreon, head over to twist.org and click the Patreon link on next week's show.
We will be back Wednesday, 8 p.m. Pacific time, and again 5 a.m. on Thursday, Central European time, broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, want to listen to us as a podcast? Perhaps while you try to make your own candles that smell like your hands? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twist.org, where you can also sign up for a newsletter of sorts.
Yeah. You can also contact us directly. Email Kiki at Kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be pulled via static electricity out of the universe. We'll never hear it, or see it, or read it. It'll be gone. The static electricity just, it's gone.
Or you can ping us on a Pulsar, where we are at TWIS Science, at Dr. Kiki, and at Jacksonfly, as well as at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week